Hello, viewers. You're welcome to our lecture series on biological techniques. Today's lecture will be on the techniques in entomology and arachnology. Entomology is the study of insects, while arachnology is the study of arachnids. Examples of insects include the wasps, the bees, the butterflies, the mosquitoes, and the grasshoppers, while the examples of arachnids are the spiders, the mites, and the scorpions. Insects are distinguished from the arachnids by possessing three body parts: the head, the thorax, and the abdomen. The arachnids have two body parts: the cephalothorax and the abdomen. The insects have three pairs of legs. They are also known to possess wings for flight. The arachnids, on the other hand, have four pairs of legs and lack wings. The insects have spiracles for respiration, make use of spiracles for respiration, while the arachnids make use of hook lungs for respiration. The insects possess antennae, while the arachnids possess palps instead. The insects possess compound eyes, while the arachnids possess usually four pairs of eyes. Now, we want to look at specific methods for the collection of insects and arachnids. Insects and arachnids are among the most diverse groups of organisms. And because of their diversity, the methods for their collection also is very diverse. Now, the collection of insects and arachnids is the method for collection is determined by the habitat and also the purpose for the collection of these organisms, the reason why these organisms are collected. The methods used to collect insects and arachnids are dictated by the ultimate goal of the samples collected. Insects may be collected as a hobby for personal enjoyment of their diversity and beauty. They may be collected in conjunction with school courses on biology or entomology, usually when they want to use these insects or arachnids for practicals in the biology lab. They can be collected for that purpose. Specific insect groups may be sampled to assess or measure biodiversity to help identify appropriate areas to be included in reserves. Aquatic species may be used to detect changes in water quality. The pest species may be sampled to assess presence or absence or abundance in order to determine whether control measures are necessary. Specific groups or species may be collected to acquire material for biological, physiological, ecological, molecular, or systematic studies. Now I want to look at the basic equipment for the collection of insects and arachnids. Which equipment do you need to collect these organisms when you want to collect them? The equipment to collect insects and arachnids include: 1. Forceps. The forceps are used for the collection of insects. We also need vials containing alcohol or other preservatives. The vial is also needed as an equipment for the collection of insects and arachnids. We also need killing bottles of various sizes. Can be improvised by using mayonnaise bottle if the normal killing bottle that's produced specifically for this purpose is not available. Then small boxes are also required. Then small envelopes. The small envelopes are used for temporary storage of delicate specimen and or gel caps for tiny specimen. They also need aspirators, one or more aspirators. Now this is an aspirator that's used for the collection of small insects. Then absorbent tissue for use in killing bottles and aspirators. We also need notebook and writing equipment for jotting down notes and label data. and a strong knife. The strong knife is used for opening galls, seed pods, weak twigs, and so on, and a pair of scissors for cutting labels. We also need uh, small, fine brushes. Usually, the camel's hair is known to be the best, and the small, fine brush used for picking up minute specimens. To do that, you moisten the tip, and then tiny specimen will adhere to it and may be transferred to a killing bottle or vial. So small brushes like this one on this screen can be used for the collection of insects. We also need bags for storing plant material, rearing material, or belly samples for collecting much plant material. A botanist's basket or tin box is advisable. Then hand lenses are also required, particularly to collect minute insects and arachnids, for the collection of minute insects and arachnids. Now I want to look at the collection methods. What are the methods for the collection of insects and arachnids? The methods for the collection of insects and arachnids include the following. We have the hand collection, we have the use of aspirator or pewter, the use of collecting nets, and the collecting nets include the aerial nets, the strip nets, and the aquatic nets. Then the beating sheets are also used to collect insects, as well as knockdown sprays. We have other methods for collection of insects and arachnids, which include the use of extractors, and the extractors that are used include the Bellis funnel or the Tilgrim funnel, as well as the Magzaski Winkler selector. We also use baits and refuges, and sieves and shifters, as well as the use of traps. The traps include the pitfall traps, the light traps, the yellow band traps, the sticky traps, the paper band traps, butterfly traps, the pheromone traps, the suction traps, the window pane or interception trap, as well as the Malleus trap. And then there's also the rearing technique for the collection of insects and arachnids, which includes rearing in cages, rearing in aquaria, and rearing in emergence boxes. Now I want to look at the first method for the collection of insects and arachnids, which is hand collection. Some slow-moving and uh, non-toxic insects are usually collected by hand. For example, this small uh, this small insect that's associated with bitter leaves, bitter leaf weevil, can be collected with the aid of the hand. Usually, it's advisable to put on hand gloves, but where the insect is known to be non-toxic, bare hands can be used to collect such insects from sight. Then we also use aspirators to collect insects. Now, what is an aspirator? An aspirator is a suction device for collecting small insects which are difficult or hazardous to capture with the fingers or with an insect net by sucking them into a container. The insects are being sucked into a container. Specimens are collected by sucking on the end of the gauze-covered pipe while holding the end of the other tube close to them. Now, this is an aspirator. The aspirator consists of two pipes, one that has a mouthpiece that's being sucked, and the other part that's being held close to the insect. Now, it also has a vial, the vial. Now, how is this aspirator being used? How is this aspirator being used? Now, the mouthpiece is being sucked by the collector, and then the other end of the aspirator, the other tube, is held close to the insect. And then when the mouthpiece is being sucked, the insect goes in through this pipe into the well, and it is being preserved for further studies. Thank you very much for watching. We want you to continue to subscribe to our YouTube channel for more important videos.